So I'm going to talk about some previous work and some future directions evaluating microbial products for soybean production. My name is Mallory Choudoir. I'm an assistant professor in the Department of Plant and Microbial Biology, and I am also soil microbiome extension specialist. So I guess raise a hands for anyone who's seen me speak before, or is this new? All right, okay, a few of you. Nice to see you again, and for those of you who I haven't met yet, hope this is interesting and gives you a new insight on some of the things that are happening in our soils and in our cropping systems. I'm going to describe the role of microbes in soil health and crop production, those activities that are often harnessed for the development of agriculture biological products, so biological amendments, and then briefly summarize the results of our 2023 microbial product trials in soybean. Okay, so microbiomes, which are the collection of all the microbes in a given habitat, and these communities really drive beneficial agroecosystem functions, right? These are the living things that are doing the stuff in the soil and interacting with plants. So microbes drive soil organic matter decomposition, nutrient cycling, really are transforming below ground pools of carbon, nitrogen, and phosphorus. They enhance water and nutrient uptake by plants through the creation of soil structure. And then there are a number of direct and indirect interactions between microbes and plants, which promote growth and can also control pest and pathogen pressure. And then some sort of broader activities that expand across geographies related to the control of greenhouse gas emissions, so really in controlling the flux of carbon between the terrestrial biosphere and the atmosphere. Sort of honing in a bit on the activities that microbes provide to plants, this kind of falls into 5 categories of plant growth promoting microbial functions. 1, microbes expand the genetic capacity of plants, right? We're talking about soybean genetics a few minutes ago, but this picture on the left shows the number of microbes that plants interact with during their lifetime. Like a lot, like thousands and thousands of microbes in these different habitats, the soil, the rhizosphere, the phyllosphere, and this literally expands the genetic capacity of a plant to not just be the plant, but the plant and the microbes, and all of those ecological functions that those microbes carry out. Improved nutrient and water access, right? We talked about in the previous slide, plant growth and development. And also, microbes increase the stress tolerance of plants and also contribute to disease suppression, again, in both direct and indirect ways. Increasing nutrient use efficiency, right? Some of these relationships are really well understood, such as the relationship between rhizobia living in the nodules of legumes and soybeans, right? So this is a very clear beneficial relationship. Bacteria provide the plant with nitrogen, and the plant provides the microbes with carbon, right? Both are happy. And then in other habitats in the soil, microbes can also improve nitrogen use efficiency and also phosphorus use efficiency, right? So microbes, again, are cycling nitrogen through different transformations and really are the drivers of the ratio between the nitrogen inputs in fertilizer and outputs. So microbes are the controller of nitrogen use efficiency. Microbes also produce phytohormones. So here, it's really challenging to read, but basically this is a single microbial species and all of the plant phytohormones that this microbe can produce, right? So this ranges from auxins, cytokinins, different acids, jasmonic acid, right? So all of these hormones that we know related to plant development and disease suppression are directly produced by microbes, right? So this is one of the reasons why microbes mediate abiotic stress tolerance. So across a number of abiotic stressors, drought, cold, salinity, microbes, the association of plants with microbes allow these plants, both domesticated crops, but also wild plant varieties, to establish and thrive in these stressful situations. And then, finally, microbes can also mediate disease suppression, both in very direct ways, like producing an antibiotic, right, that targets a pathogen, and also in these more complex ways. So microbial communities are behind this phenomena of disease suppressive soils, right? So we have soil microbes that can suppress disease, and then others that are conductive, right? So this is a complex phenomena that we don't quite understand, but it's something to do with the microbial communities. Really, if it's useful to a plant, a microbe has already invented it. Deviating a little bit from soybean here, but this is an image of a teosinte ancestral corn variety in Oaxaca, Mexico. This is from a few years ago, and this mucus, it's produced by the plant, so it's this like gunk, like literal plant snot, that's really high in carbon, and microbes live in that. And in this mucus are free-living nitrogen-fixing microbes, not colonizing nodules, right? So it's not a legume, it's a cereal. And these free-living microbes are providing nitrogen to the plant. So this product, right, is now what has become the Pivot Bio's proven varieties of microbes, which are free-living nitrogen fixers that they're developing for cereals. Okay, so for all of these reasons, all of these activities, microbes and other biologicals represent this rapidly growing market. So here are just some of the companies that are involved in developing products in this space. And in the next few slides, we'll go over some of the categories of these different microbes. But just to point out that this market is massive now. The global market value this year is $6 billion, and then in the next 10 to 15 years, this is, will continue to expand into many billions and billions of dollars. So lots of opportunity and also lots of challenges of navigating this massive market. So I want to because of this vast market, just take a few slides to sort of go over what these products are, right? So, we can classify them by source. So microbial versus natural product. A microbial product is a living microbe, right? So the product itself is a living cell versus a natural product. A natural product is anything that is produced by a living organism, but not the organism itself. It can be a little tricky because if you have a microbial product, it's likely that that product is producing a natural product, but you can also just apply the natural product itself. So here's a number of natural products produced by living organisms. They can be microbes, they can be plants, and these include a number of antibiotics, right? So natural products are also what we give ourselves in the hospital when we step on a nail or something. And just to, in the world of drug discovery, a natural product is a single molecule that has an activity, right? So that's a specific definition of what a natural product is, but labels can be challenging to digest. So natural product can also be put as opposed to synthetic, right? A natural product naturally occurring versus synthetic, which is man-made. I will say again, products are so tricky to categorize, so there are certain products that are naturally occurring, so a lot of seaweed extracts. But the way that that product is produced then sort of floats into the synthetic category because of the methods used to create what goes into the field. So again, some of these are not as clear-cut as you might think they would be. And then, right, importantly, organic is not necessarily a natural product, right? So there are organic rich amendments, which are fertilizers, which again can be organic, so naturally occurring versus synthetic man-made. So again, all of these are organic soil amendments, but would fall under the realm of fertilizer versus natural product. The definition of these biostimulant was sort of recently defined by the USDA and EPA. So biostimulant is a substance or a microbe that, when applied to seeds, plants, or the rhizosphere, stimulates natural processes to enhance or benefit nutrient uptake, nutrient efficiency, tolerance to stress, or crop quality and yield, right? So a lot of those activities that we talked about. That's what a biostimulant is. And again, right, there are some interchangeable terminologies between microbial inoculants and biofertilizer, which again is really confusing because a biofertilizer can be a natural product or it can actually be a fertilizer, which is nutrient content that the plant directly uptakes. Conversely, biopesticides, so this definition is very precise, right? So anything that has biocidal activity is highly regulated by the EPA, so this is more straightforward. So biopesticides are anything, including naturally occurring substances, that kills pests. So that can be biochemical, it can be microbes, or plant-produced substances that just kill pests. Okay, so that was a very brief run-over of some of these classes of microbial and biological amendments, and I'll be putting together some resources to come back to that content on our new soil health extension portal, which I'll share in a bit. Okay, so briefly before lunch, I want to talk about our results of our commercial microbial product field efficacy evaluation. And this was sponsored by the North Carolina Soybean Producers Association. And we're excited to continue our work with them moving forward into this next season as well. Okay, so for this last year, the goal of this research was a simple, do microbial seed treatments increase soybean yield? And then does this vary across production region? And as a microbial ecologist, microbiologist, my second question was, does the presence of this living microbe on the seed shape the rest of the community that assembles with the soybean? And again, does that vary across region? So here are the three products that were included in this study last year. You notice one of them is still blocked out as a overabundance of legal caution and also out of respect for the industry partners that I worked with. So the identity of that product will be available when the report is released on the portal very soon next month. These products range, they're all microbial biostimulants, ranged from fungi, so the Endoshield ST is a single fungal species, BASF Vault IP+, which is actually a combination of two microbes, a rhizobia and also a bacillus. And then this third product, which is a complex mix of different bacteria and fungal species. And then, of course, a non-treated. So the application rate was done by the product recommendations. We standardized the volume of the application rate. And then, of course, best practices stored as recommended, and then also planted within 1 week of treatment. We treated 2 different varieties to capture different maturity groups. And we installed these trials with the help of Ryan Heiniger and the official variety testing program in Union County, Beaufort County, just down the road. I'm pretty sure I passed the field on my way here, and then in Sampson County. So 3 microbial treatments plus a non-treated across 2 maturity groups. I'll share the yield, and this was randomized plots with 4 replicates each. We also have leaf nutrient data, soil biogeochemical measurements, and then in the near future, microbial analysis of the roots rhizosphere in soil. Early development leaf nutrients differ between locations, but not between microbial treatment or variety. So here we have Beaufort on the left, Sampson on the right. It was raining cats and dogs when we went to Union County, so we lost those tissue samples. So we have NPK percentage in these columns, and these are box plots, right, which show the spread of the data. The line in the middle is the median. So this, again, we see a significant difference between nutrients in nitrogen and potassium, but not phosphorus between these locations, but no difference between the microbial treatments and the control. And then just to say, even though there are significant differences, neither of these extend into the toxic or deficient nutrients. Yield also differs between location, but not by microbial seed treatment. So here again, we have all 3 locations, Beaufort, Sampson, Union, the 3 treatments plus the non-treated. The dashed line is the mean yield per location. So we do see significant differences in yield between locations, but not within location between the control and the treatments. Breaking this up a little bit more, within each location, we did see that yield differs by variety, but again, not by microbial treatment. And all of this information is probably, arguably more readily digestible within the context of all of the OVT data. So this is consistent, the varieties and the trends between locations is consistent with the larger OVT variety trials. So would recommend following up in a broader context with that data. So in summary, location and variety determine yield consistent with OVT results. No significant differences in yield between microbial treatments and the non-treated. However, looking at this data, I will say that despite the robust design, the small sample size and the large variation in yield even between a single treatment really makes robust, statistically supported inference difficult. So we would really need to see a big difference in yield to find a statistical difference here, which we did not. So that is the summary of last year, microbial data to be determined. Excited about that. But moving forward, we'll be expanding acreage and plot replication for some stronger inferences. And then also I've been in some conversation. I'm planning to target some products that hopefully will address salinity tolerance out here in this region. So check out our new soil management portal. Soon there will be another column on here that will have the results of the biological amendments from last year, and then moving forward will be posted there. And with that, I'm happy to take any questions, or feel free to reach out at a later date. Thank you.